Okay, what time is it? Almost time. While we're waiting, if you have printed up your Thursday lab schedule, you might want to get it out now. All right, it's 1 o'clock. Because this is what we're going to be doing today. Going to start out by getting you guys all centered in on anatomy lab. I do have to take attendance and do the last add codes. And then we have to do the first lab on histology. And if you're wondering what histology means, it means microscopic anatomy. And if you recall the hierarchy of biological organization there at the bottom, I said on Tuesday that lab this week is on these 3 levels. Does that look familiar? Okay. All right. Okay, let's first talk about anatomy lab and ERT. Probably starting in week 3, I will be doing your labs, the initial part of it anyway, downstairs in the anatomy lab, because I want to utilize the models. Well, first it'll be bones, and then eventually models. I didn't do that the second half of last semester because it was, again, a real sudden thing, and I was working on physiology labs actually instead of doing everything for anatomy lab. But this semester, what I want to do is I want to actually go over the lab. Everybody got to mute yourself. Bottom left, make sure you're muted. Okay. As I'm going over the lab PowerPoints, I want to actually have the models out in the camera so that you can actually see what the full models look like instead of just pictures of them. Okay, so, but for the first 2 weeks, we don't have models, so I'll be doing your lab in my office, which is a little nicer for me, okay? Now, if you look at the lab schedule, you can see the various lab topics. If you have your laboratory objectives, open it up, and that's your best friend. I'm going to keep calling it your best friend. Turn it to the first page, which is the table of contents. Here you can see with the lab schedule that the pages for the laboratory objectives corresponds to the right column and the table of contents. And then the lab topics are the same lab topics in the table of contents. And we're just going to go right down in order. Now, what you will also notice is that there is one lab quiz 2 weeks from today that covers what I'm covering half of it today. And then I'm covering the second half next week. Then the following week is a lab quiz on it. That is our first graded work for the semester. I forgot to tell Tuesday lab too. I'm going through the Proctorio training tomorrow. And so next week in lab, we're going to have a test quiz, a fake quiz to run through Proctorio. So hopefully we will have iron out the bugs, and I'll probably do that like around 3 o'clock during your lab period, okay? And then after that, we get into lab exam 1 material, which is on bones. There are 5 lab exams. And if you notice, I'm going to finish covering the skeletal system, and then you got 2 weeks before we actually have the lab exam. And that is to give you time to study and drill and practice spelling and to have it down, okay? Because remember what I said on Tuesday lecture, the secret to passing this class the first time is passing lecture exam 2 and then kicking ass in lab. You do that, you've taken this class once, okay? Just like the other lecture schedule and the course syllabus, I've done it the same way with ERT, things that we usually did live. I want you to see what it looks like, but it has a line through it that we can't do remotely. And then also at the bottom or near the bottom, you can see the actual Zoom link for lab next week, and that Zoom link for next week will be good for the rest of the semester. I will be sending out an announcement in Canvas that has all the links, lecture, lab, and my office hours. Those are the links for the rest of the semester. I had to use this separate link this week because I had unregistered students on the wait list still needing to have access, okay? So we got 1 lab quiz, 25 points, 5 lab exams worth 50 points, and this is what the live lab exam form looks like. There's 25 stations, A and B, 1 point each. That's your 50 points. And like I was saying in lecture Tuesday, each lab exam has 10 points of extra credit. There are 60 possible points for a 50-point lab exam. That's why you want to kick ass. Now, in Canvas, you don't have a form like that. It's just all fill-in questions. Please use small case letters only unless you're using somebody's name. All right? On the very bottom are some of the lab policies that are just considered part of the syllabus. Again, I told you that lab is only about 2 things: visually recognizing structures and spelling them correctly. And the first penalty, you see what happens if you don't spell it correctly. You only get a half point. That means that you only are missing a couple letters here and there, okay? If I can't tell because it's so terribly misspelled that you even knew the answer, then you're not going to get any points. But if you just, switch a couple of letters, you get half credit for that. I also said on Tuesday's lecture that you could get 60 possible points when you take the lab exam when you're supposed to. If you take it with, you know something's happening on Thursday and you don't want to lose the extra credit, you have to email me with enough notice and take it with my lab on Tuesday. If you take it at the same time as other people, then there's no penalty. But if it's at a different time and it becomes a big Canvas pain in the ass, then you just don't get the extra credit. That's the only thing. So you get none of those. So that's the 10-point penalty. No EC, no extra credit. Really, for the other penalties, if you don't, especially after this week, I'm not going to provide any recordings. If you miss lab, you're really only screwing yourself. So I don't know if I can assess these other penalties. I don't know if I'm going to, but you should be aware of them, okay? You'll be tired when we take the lecture exams in the morning. You still got to do lab that same day. This is one of the things your programs want you to get used to, and that is being able to perform when you're tired, right? Nurses have got to work even if they're tired. And the only way you learn that is if we make you do lab after lecture exams. So that's why it says you can't miss a lab on an exam day, okay? Any questions on the lab schedule? Okay, let's see, where are we? Lab points, let's see, so 25 points, 5 times 50, 275 lab points. You got the schedule, you got the policies. All right, I'm gonna go ahead and take attendance now on my sheet. What you can do is you can go ahead and look over the PowerPoint and turn to page 5 of your laboratory objectives and look that over. Anybody here who has not received an add code who wants to add, everybody hear this. If you are still wanting to add the class and you haven't gotten a add code, email me now. So that way it'll be really easy later on in the lab. I'll just reply with the ad code, okay? But let me get doing this now. Maria, you're in there twice. That's because there's two, I got two people in the same name. Okay, Maria Herrera, Maria Herrera. All right, that's done. Okay, again, if you, I got some here. Okay. Cynthia, we will talk about how we're going to work this with the switching labs. And I got one request for an ad code. I think I got it, everybody already added. Yeah. Okay, I'll send that in just a bit. Now let's go ahead and turn to page 5. Well, let's first turn from the table of contents. Then we got a purpose statement there, page 3. Those are my former colleagues, and I'm the only one left. Okay, page 4, that's topics that we covered in lecture on Tuesday, including the linings of the cavities, posterior cavity has meninges, anterior cavities have mesothelium. Does that look familiar? Covered that in lecture. But when you look at the lab schedule, there is really nothing on page 4 I'm going to test you on in lab. That's for lecture. The lab quiz is histology, page 5, and the integumentary system, page 6. That's it for the lab quiz, okay? And it's just going to be 25 blanks. Half of it is from page 5, half of it is from page 6. But today we're going to cover page 5. Now, one of the things that I wrote on the whiteboard, and here it is on the clipboard, and that is, with all the structures of the human body, there are only 4 tissue types. That's it. Out of everything that we're going to learn this semester, all of it is only 4 tissue types, and you can see that on page 5. Okay, let's get going on the lab PowerPoint here. It's kind of a bummer that in ERT, you're not going to be able to actually look through microscopes. We actually only have 2 labs where we look through microscopes in the live anatomy class. And for a lot of students, this is the only experience they have with working with microscopes before they take microbiology, micro. And now you're not even going to have that. So hopefully you'll have some training before you get into microbiology. Okay, here's the secret. I'm going to start, and again, I don't teach anatomy. I use anatomy to teach you how to be health science students. What you want to start learning how to do is organizing content. This class, it'll seem like it has a lot of content. Actually, for the classes that you are going to have to take later, it's not that bad. But this is a good type of class to train you how to organize content, because there are going to be classes later that have more, okay? And one of the ways to do this is just always keep content in order. This is one of the reasons why I did not, as a student, like flashcards. I know a lot of students do, but I got to tell you, flashcards, you can throw it up in the air, pick them all up, they're all out of order. And that's exactly what happens in an exam. Just like all those flashcards can be jumbled up and swim around in different orders, that's kind of what happens in your brain. It just goes swimming around. It's there somewhere, you just can't find it. Well, if you keep the content, and it says it right on top of the slide, organized, it has a better chance of not going swimming on you in your head during the lab quiz. There are 4 tissue types. Learn it in order, drill on it in the same order, and it won't go swimming on you during the lab quiz. Look on page 5. It's epithelial tissues, connective tissues, muscle tissues, nervous tissues. 1, epithelial tissues. 2, connective tissues. 3, muscle tissues. 4, nervous tissues. Everybody got it? Keep it in order. Now, on the very top, where it says epithelial tissue, it says line or cover internal or external body surfaces. Epithelial tissue is a lining tissue, okay? So it's cells forming a tissue that line things. It can line things on the exterior, like your epidermis is epithelial tissue. It's lining your whole body on the outside. Your blood vessels on the inside are lined with epithelial tissue. It's called endothelium. It is lining the inside of structures as well. So these cells that are lining things, I would circle that or write that down, lining tissues. The cells can either be a single layer or it can be 2 or more cells thick. If it's a single layer thick, it's called simple. If it's 2 or more layers, it's called stratified, which means layered. Notice it is the yellow word. Then epithelial tissues are classified by the shapes of the cells on top. We'll cover this more later, on the free surface. If they're flat, wider than they are tall, it's called squamous. If they are about as tall as they are wide, It's called cuboidal, like a cube. It's not really cube shape, it's six-sided, hexagonals, but it's just as tall as it is wide, approximately. If it's off a little bit, don't get your underwear in a knot, it's still cuboidal. It's approximately as tall as it is wide. And then you have cells that are taller than they are wide, like a column, that's called columnar. That's the purple word. Now go to page 5 and look at the 6 epithelial tissues you need to visually identify. Do you see yellow and purple words? Do you see why I cover this first? So that way the words actually have meaning, okay? Now there is one of the tissues that does not have any yellow or purple words, and that's transitional epithelium. Okay, and that's, well, I'll explain it later. It's constantly changing. All right, now I got these slides from my lecture PowerPoint because we do cover tissues in lecture, but I kept this whole slide because even at the very bottom, this is a note of visual recognition. You guys got to kind of get that. Lab is about visual recognition. The notes that you take should be about visual recognition. And in epithelial tissue, you never see blood vessels, and that's avascular. And since it's lining things, it's always stuck to something. And what it's stuck to is a basement membrane. So yes, your epithelium, your epidermis is stuck to a basement membrane. Okay? All right. So if you want to know you're in epithelial tissue, since it is lining something, this is what you're going to see. First of all the slides for epithelial tissue will be stained. These cells don't look this way in real life. It only looks this way when we stain the cells. And the nuclei pick up the stain, and that's what these purple balls are. You're going to see a ton of stained nuclei really close together, and on one side, they come to an abrupt end. Everybody see this line right here that I'm moving? That is the basement membrane. On the other side is nothing, since this is a lining tissue. This slide was taken from a trachea, and this is right here is where the air is passing through, the free surface. Epithelial tissue is between the basement membrane and the free surface. Basement membrane, free surface. Basement membrane, free surface. That's epithelial tissue. Okay? Now this is key, because if you see this, you are not worried about tissues 2, 3, and 4. You know you're in number 1 epithelial tissue if you see a basement membrane and a free surface. Now let's go over the 6. Here, and this is also, remember that epithelial tissue can also line the inside of tubes. This is not a blood vessel, this is a kidney tubule. Okay? Look at these arrows. This purple line right here is the basement membrane. The hole in the middle of the tube, this is a tube in cross-section. It's like running like this, pointed right at the camera. You cut it, boom, it looks like that. So the hole in the middle, it's called the lumen, L-U-M-E-N, is the free surface. If that's the case, this has got to be epithelial tissue. Now look at that picture. How many cells thick is it?
1.
1. What's the yellow word?
There.
Simple.
Simple. Look at the previous slide. I guess 2 slides ago, or right here. 1 cell layer thick, it's simple. Are these cells approximately as tall as they are wide? Yeah, this is a little wider. This is a little taller, but they're all approximately as tall as they are wide. Again,
Yes.
underwear and a knot about that. What is the purple word if the epithelial cells are as tall as they are wide?
Cuboidal.
Cuboidal. This is a simple cuboidal epithelium, okay? Now, I'm going to show you this here. See if you can see that in the picture. If I do show that on the lab quiz, these are your only two choices for a full point. You have to say simple cuboidal, but after that, you only have two choices, epithelium or epithelial tissue. That's what you need for the full point. Maybe screenshot this if you want. If you say simple cuboidal epithelial, that's only a half point. If you say simple cuboidal epithelium tissue, that's a half point. These are your only two choices.
But we have to write both or just, I didn't get it.
One, either or.
One of them, either or, okay.
But the whole thing. How's that? Does that make it clear?
Yes.
Okay.
Yes.
All right, let's go over the 6. One cell layer thick and the cells are flat. By the way, remember mesothelium is all the peritoneum, pericardium, and pleura in the anterior cavity. All that is simple squamous epithelium, mesothelium. It's found other places as well. You look at this picture and you go, oh man, that looks like multiple layers. No, it's this up here looking from on top. If you look down on this picture on top right here, it looks like this. Everybody got it? You know, I did get these slides from the lecture PowerPoint. You don't need to know all this. You want to use these notes from the lecture PowerPoint for things about visual recognition. One cell layer thick and they're all flat. We will see some of this lining some kidney tubules, and we will also see it aligning what's called the Bowman's capsule or glomerular capsule in the kidney as well. But one cell layer thick, thin cells, really flat cells. All right, let's look at this slide. This is stratified squamous. Next week, I will contrast this with both keratinized stratified squamous and non-keratinized. Your epidermis in your skin is keratinized, and when that happens, when you see that, there are no stain nuclei at the free surface. When it is non-keratinized, like inside the lining of your mouth, like your tongue, you will see nuclei, these stained purple things, all the way up to the free surface. That means it's non-keratinized. Now look carefully at this picture, and now I'm going to go back to the purple word. I said it briefly about the purple word, that it is only the cells on top, only the cells at the free surface. So when we go back, now go back to this slide here, to the purple word, and write somewhere off to the side, this is only for the cells at the free surface. Cells underneath don't matter. Only the cells on top at the free surface for the purple word. Because look at this picture. Down here at the basement membrane, and that's the thing about basement membrane, it's where the stain nuclei come to a sudden line and stop right here. Everybody see the basement membrane? Look at some of these cells. Some of them are columnar. Most of them are cuboidal. They're still cuboidal. They're still cuboidal, but now they're starting to flatten out. And by the time they get up here, they're all flat. At the free surface, they're all flat. That's why this is stratified squamous. It's only about the cells on top. The shape of the cells down here do not matter. Are we clear? Here's that simple cuboidal epithelium we saw earlier. You say, well, gosh, number one, that looks like it's stratified. Yeah, no. Oops. This down here is one kidney tubule. This right here is another kidney tubule. Each is simple cuboidal. Each is only one cell layer thick. Right here where they touch, both tubules are sharing a basement membrane. Sometimes in a basement membrane, you see a capillary. And this is a red blood cell in this capillary. But this is not stratified. This is a simple cuboidal epithelial kidney tubule. And down here at the bottom, this is a simple cuboidal epithelial kidney tubule. Does that make sense? Does that make sense? Now, why does this look different? Look in that tiny picture of me on the top right. This picture, the kidney tubule is running like this. This is what is called longitudinal section. In this picture, the kidney tubule is looking like this, looking straight at you. This is cross section, but it's the same tubule. Boom. Or boom. Everybody got it? Are you glad this is being recorded? Here's the trick. Even if it is recorded, Zoom can still have a glitch. Stay engaged. All right. Transitional epithelium is next. It is actually stratified, 3 to 8 cell layers thick, and you might be tempted to give it that yellow word, stratified. But then you look at the free surface for the purple word, and you find squamous, cuboidal, and columnar. And you go, well, what do I call it? Well, then you've got to remember there is no purple word. You have multiple cell shapes at the free surface. That's down here at this bottom box. Yeah, it's stratified. You're going to be tempted to give it the yellow word, stratified. Then you go to the purple word. There is no purple word. This is transitional epithelium. Multiple cell shapes at the free surface. What does transitional mean? Alon, you know what the word means.
That it's changing to another tissue.
It's not changing to another tissue, but it's changing shape.
Oh, okay.
Because look where we find it, urinary system. Think about your bladder. Just before lab started, because you didn't want to have to turn off your camera and go to the bathroom. You went bathroom beforehand. Well, when you do that and you empty your bladder, it goes like this. And all the lining epithelial, transitional epithelium becomes columnar. But then, just like me, and because we're not in a lab where you can't have any food or drink, now you can because you're at home, you're having caffeine to stay awake. Caffeine is the diuretic, and it's increasing your urinary output and producing your bladder stretches to more cuboidal. And after a little while, you're going to say, Miyahara, can you take a break? Or I'm just going to have to turn off my camera and go to the bathroom because it's so stretched out, they're all squamous. And then you go to the bathroom and you piss like a racehorse and it goes cuboidal and columnar. Do you see why it's called transitional? Well, when you have this type of tissue, because of the elastic connective tissue underneath it, whenever you cut into a bladder wall to get a slide of transitional epithelium, it results in multiple cell shapes at the free surface. That's how you can tell it. That is a good thing to write down as a note of visual recognition. Okay, simple columnar epithelial tissue. You can see it's one cell layer thick, simple, and the cells are a lot taller than they are wide, and you can see it right here. This is the predominant tissue lining of the GI tract. This slide was taken from a small intestine. What you want to write next to this slide is simple columnar epithelium. All the nuclei line up in a row. Do you see this right here? Do you see the purple oval shapes? Those are the nuclei. They're all right in a row. Okay. These are not the tallest epithelial cells your body makes. They're pretty tall, but they're not the tallest. The tallest epithelial cells your body makes is this, pseudostratified ciliated columnar epithelium. If I were you, I would say it like this, E before U, pseudostratified ciliated columnar epithelium. Again, you have to either finish those three words with epithelium or epithelial tissue. That's your only two choices.
And we have to spell that correctly for our lab quiz?
Yes,
Oh, that's gonna be
otherwise you
fun.
lose a half point. That's on the bottom of your lab schedule.
That's going to be fun.
Yeah, E before U, pseudostratified. Say it out loud. E before U, pseudostratified. The most common lost half point of the lab quiz. Okay. This is the predominant tissue lining of the respiratory tract. You look at this thing, okay, now I'm going to explain to you why this is your best friend. Because there are actually 8 epithelial tissues. Your best friend only has 6. Are you following me? This is the limiting list. There's actually going to be just a couple of exceptions later on in the semester, but in a basic nutshell, if it's in here, I can ask it. If it's not in here, I can ask it. Your body actually does have stratified columnar. Because when you look at this picture, look at this. Oh, it looks stratified. You look up here at the free surface, you can see that's all columnar. And you might want to go write stratified columnar. It can't be stratified columnar. Do you see stratified columnar on page 5? No, that means I can't ask it. If it looks like stratified columnar, it has to be this, pseudostratified ciliated columnar epithelium. This is actually the tallest cells your body makes. This is a good illustration of that. Here's simple columnar. Cells are taller than they are wide, but it's only so tall, so all the nuclei have to be lined up in a row. That's why you needed to write that down on that slide. All the nuclei are in a row. Pseudostratified ciliated columnar epithelium are so tall, the nuclei are aligned on different levels, making it look stratified. When look at this. All these cells, this is one cell layer thick. It is a fake stratified, which is what pseudo means, like fake boobs. This is fake stratified. Every one of those cells in that picture is on the basement membrane. It is one cell layer thick. And by the way, these little hairs that the arrows, these black arrows are pointing at, these are the cilia. That's why you need this middle word. Pseudostratified ciliated columnar epithelium. This is showing you all 8, and on this slide, for the lab quiz, go ahead and put an X through this one, and put an X through this one. Both of them are kind of rare, both of these. We will cover these in lecture, but you are not responsible for them for the lab quiz. Why? Because in slides, and you're not trained in reading histological slides, this looks a lot like this. But now that you put an X through this, you know that if it looks like that, it has to be this because of your best friend. Any questions? Okay, that's tissue type number 1. epithelial tissue. Everybody listen to me. The thing you're looking for tissue type number 1 is a basement membrane and a free surface because it's a lining tissue. Tissue type number 2 is going to be your most challenging. The reason why is because these tissues look all different, highly diverse. But read what it says on top of this slide. These are living cells living in non-living stuff we call matrix. Because it's living in this matrix, it is separated by matrix. These cells are separated by stuff. They are not close together, not like epithelial cells. They are separated, but that's what you're going to look for. Now, there is one connective tissue that doesn't apply to this, but it is so obvious, I don't think that's going to be a problem. Okay? So this is showing you the cells, the living cells. That's for lecture. And the stuff that it's living in. And that's matrix. Okay? Now, what I want you, the reason why I have all this information in here is I want you to write a couple things on these top 2 diamonds, because this does relate to how things look. Where it is relevant, collagen fibers are going to stain pink. I would write the word pink next to collagen. Okay. Where it is relevant, elastic fibers are going to be stained black. I would write black next to elastic. Okay. Now, let's go through the list, and then we'll take a break after we get through connective tissue. Are you guys ready? There's actually 6 types of what we call connective tissue proper. Of those 6, on your best friend, there are only 3. Here's 2 of them. Okay? You do have to learn all 6 for lecture, but for the lab quiz, you only have to learn 3 out of the 6. Here's 2 of them. Look at loose connective tissue. You can also call it aurelian connective tissue. Which one's easier to spell? Yeah, loose. That's what I would do, loose connective tissue. You're not impressing me if you write out aurelian. Don't write both of them like that's going to impress me. Just write one and go with the easier one. Loose connective tissue. Oh, by the way, let me go to the laboratory objectives, page 5, and I'm going to give you the words you need to include to get the full point for the connective tissue. I already did the thing with epithelial tissue. You got to just have epithelium or epithelial tissue after it. Okay, you guys ready? Paying attention? First one. You need 3 words, like what it says on the slide, loose connective tissue. Next one, just 2 words, like it is on the slide, adipose tissue. You just need 2 words. The one after that, 4 words, dense, regular connective tissue. Next one, 2 words, hyaline cartilage. Next one, 2 words, elastic cartilage. Next one, 1 word, fibrocartilage. Next one, 1 4-letter word, bone. Next one, 1 5-letter word, blood. That gets you the full point. All right, let's look at loose connective tissue, 3 words. If you look at that picture, you see a whole variety of cells that were listed on the previous slide. And it looks like someone just took all this diverse cells, connective tissue cells, and went blap. Then you take some fibers and you just go blap. And when it hits the screen, it looks like this. You got fibroblasts, you got mast cells, you got macrophages. Look at this, people. In this slide, you see pink collagen. They're thicker. Then you also see these wire-thin black elastic fibers. Did you guys write pink and black on those fibers before? There it is right there. And it just looks like a mishmash of cells with a mishmash of fibers. That's loose connective tissue. But you can see the cells are splattered, separated by stuff. Yeah, every now and then two are touching. But oftentimes when you see two cells touching, it's because they just went through mitosis, we'll talk about later. They just went through cell division. They're about ready to be split apart and separated by stuff. Adipose tissue. What I would write next to adipose tissue is this is the exception. This is the connective tissue where the cells do touch. And you can see it right here. But what I would write next to adipose tissue, not only is it the exception, cells do touch, but they are extremely thin-walled bubbles. Look at that picture. Extremely thin-walled bubbles. There are some cross-sections of simple squamous epithelial tubules that can look like this, but the bubble wall is thicker because it's a whole squamous cell. Okay? So that's what it's saying here in this lower right. To an untrained eye, adipose can look like simple squamous epithelial kidney tubules, but these are going to be thicker. Adipose is extremely thin-walled bubbles. The only connective tissue where the cells routinely are close and touching. Okay, here it is written out. 4 words, dense regular connective tissue. This is what we usually think about with connective tissue as it connects things. What you want to write next to dense regular connective tissue is that it is dominated by parallel lines. They don't even have to be straight. They can be wavy, but they're parallel. And it has very few nuclei. That's notes of visual recognition. Dominated by parallel lines, very few nuclei. Here, these parallel lines are vertical up and down, and only these little black little dashes are the fibroblast cells. The reason why the nuclei are separated is because these parallel lines are not cells. They are collagen fiber. What color do you see here? Pink. Dominated by parallel collagen fibers, and because those are not cells, there are very few nuclei. Now look at this note on the bottom right. If you are not paying attention, you might think this is smooth muscle. The difference is smooth muscle lines are all muscle cells. Look at these parallel lines here at the bottom middle picture. But all these lines are cells. So that's why you have a whole bunch of nuclei, and especially you see nuclei touching almost. That's a big hint. If you see new parallel lines with a lot of nuclei, some of them touching, that's smooth muscle. That's why it says on the bottom, it has more nuclei than dense regular connective tissue. Everybody connect those dots? Okay. Fewer nuclei than smooth muscle. All right, let's go to cartilage. All right, you just need to know from the words up here that you are going to see cells separated by stuff, and here's the matrix of stuff. The cells are called chondrocytes, and they live in chambers called lacunae. The E at the end means plural in Latin. You have 2 lacunae. You have one lacuna, you take off the E, okay? And there's 3 subforms of cartilage. They are all listed right in the middle of page 5. And this is how you're going to tell that it is cartilage. Pay attention. You see the light halos around the cells? You see these are quite large, these halos in elastic cartilage. Do you see the light little ring around the cell? Everybody see that? That's the lacuna. Look at this middle picture. Some of these look like an eye's looking at you. Do you see this right here? The cell is the purple thing in the middle, the chondrocyte. And then that clear ring around it is the lacuna. Everybody got it? So you got to recognize lacunae. There's only 2 connective tissues that have lacunae. One is cartilage and the other is bone. But the lacunae in bone is really obvious because they're set up in these tree rings. It looks like a cut tree trunk. If I show you a slide of bone, it looks like a cut tree trunk or several cut tree trunks. Please do not call them tree trunks. They're a bone, all right? But these lacunae are not in cut tree trunks. This now you have to look at the fibers. Here, if you see lacunae not in a cut tree trunk, and you see black disorganized fibers, that's 2 words, elastic cartilage. If you see lacunae but not in a cut tree trunk, and you see roughly parallel, roughly, it's organized collagen fibers, pink. That's 1 word, fibrocartilage. If you do not see fibers, there actually is collagen, but it's so sparse you can't see them. So if you cannot see fibers, it looks like cells in lacunae stuck in jello. That's hyaline cartilage, 2 words. Now, I tell students that I teach defensively, which means I teach to minimize student bitching. And so if you look at that figure on the right, I would never use that in a lab quiz because you cannot see the lacunae. If I use any slide of hyaline cartilage, you'll see the lacunae. Everybody got it? But you won't see fibers, okay? Black, disorganized elastic fibers. Pink, roughly parallel, organized collagen fibers. Then here you can't see fibers. Any questions? Okay, we're almost done here and then we'll take a break. Here's that cut tree trunk. Doesn't that look like a cut tree trunk here down at the left side of this figure? This is not as good, but this is a really good one. Does that kind of look like a cut tree trunk? Its actual name is osteon, but that's not on page 5, so don't put osteon, just write bone, 4 letters, bone. Blood is really easy because it is dominated by red blood cells, and red blood cells don't have nuclei. So if you see a slide that has a bunch of pink or purple circles without nuclei and it's dominating the slide, that is blood. You will see every now and then a larger cell that does have nuclei. Those are the white blood cells, but they're far fewer and in between than the red blood cells. These little speckles down here, those are platelets. Okay, these guys here are platelets. But it's dominated by red blood cells, no nuclei. Any questions? I'm gonna come back here after the break to this. All right, go ahead and go to the bathroom, get something to drink, stretch your legs, and look at your clock on your device at 2:05. I am going to continue on with the lesson, okay? Oh, I forgot to pause the recording. All right, well, there's going to be 10 minutes of dead space. You just fast forward it. Okay, as we are gathering back, I just want to talk about a strategy, a mental approach to the lab quiz that you might want to think about. I've taught this class for almost 16 years, and I have seen students try to memorize things with flashcards, and it's just not a good way of doing it. It is better to think in an organized fashion. First, in your studying, think about things in order: epithelial tissue, connective tissue, muscle tissue, nervous tissue. And then what I would do is I would approach it like playing this game called 20 questions. Now, I know I'm dating myself. Not too many people know what 20 questions is. But, you know, it used to be when families would go on a long vacation in the car, the radio stations would even cut out once you get out of a city. Well, now everything's satellite and nothing cuts out, but everybody's doing this in car trips. You know, nobody's even interacting anymore. So that's why a lot of people don't know what 20 questions is. 20 questions is when somebody says, okay, I'm thinking of something, and the rest of the car has 20 questions to figure out who or what it is. And my youngest son just sucked at it, because whenever we were on a long ride, I said, okay, I'm thinking of something. He just like, go, is it our neighbor Zach? No, it's not our neighbor Zach. And now you only have 19 questions left. And what you really want to do with 20 questions is you want to start with big general questions and get gradually more specific. Like, is it animal, vegetable, human, or manufactured? And then your questions change depending on where you go with the first question. That's what you do with the lab quiz, especially the histology portion of it. Your first question should be, which of the 4 tissue types is it? Now, I hope you're going to take this seriously, because this is going to be really helpful. If you see a basement membrane and a free surface, it's tissue type number 1. If you see cells not close together, or it could be that one exception of extremely thin-walled bubbles, but if the cells are separated by stuff, it's tissue type number 2, connective tissue. If you see stacked tubes, these tubes are all cells, so they're touching. If you see stacked tubes, that's tissue type number 3, muscle tissue. If the question on that Canvas lab quiz does not say what tissue is this in the picture, but rather says what is the name of the cell or what is the name of the structure, that's tissue type number 4, nervous tissue. Because I'll get to it here in just a second, there are no tissues in tissue type number 4. The first 2 are cells, the last 2 are structures. And it's going to be written in the Canvas quiz. Did everybody get that? Basement membrane and a free surface. Tissue type 1. Cells not touching, or they're separated by stuff, except for adipose tissue. Tissue type number 2, connective tissue. Stacked tubes, tissue type number 3. No question about tissues. It's either cells or structures. Tissue type number 4. Look at this slide on cartilage. Say you're looking at this middle picture, and you see the cells are separated by stuff. You don't see a basement membrane and a free surface. So you say, all right, I'm in connective tissue. Now I'm going to look at this thing, and I say, I don't really see a mishmash of cells or a mishmash of fibers. Rather, I see a halo around the cell. That halo is lacuna. Oh, this is not bone because it's not a cut tree trunk. This is cartilage. Now you're getting more specific. The last questions you're going to ask is about fibers. Do you see black disorganized fibers, roughly parallel pink fibers, or no fibers? I see roughly parallel pink fibers. This is one word, fibrocartilage. I got the point. I'm moving to the next question. Everybody got it? First, get it into the 4 tissue types. That directs your next questions. This is systematic thinking, something that is going to be really important to you when you get into your healthcare programs. I'm going to instill it in you now in your first prerequisite. All right, we already covered bone and blood. Let's go to tissue type number 3, muscle tissue. These are all stacked tubes. These are all cells in those bottom 3 pictures. These are huge tubes because hundreds of cells have fused to form it. Because each of those individual cells had nuclei, those extra nuclei now get shoved off to the side. This is what is referred to as peripheral nuclei. And that's these dark stained things off to the side. Also, perpendicular to the long axis of these large stacked tubes, you see tiger stripes. Real obvious tiger stripes we call striations. In lecture exam 2, I'll teach you why they look this way. Smooth muscle, we already talked about this. You're going to see parallel lines, but all these lines are cells. So it has a lot more nuclei than dense regular connective tissue, a lot more. Cardiac muscle, stacked tubes, but these are individual unfused cells. So the nucleus, the nuclei are in the middle of these tubes. What you are going to see is some subtle striation, not nearly as obvious as this. It is much more subtle, but you will see some striations. But this is what is going to be key. If I'm going to give you a slide of cardiac muscle, you will see intercalated discs. What do intercalated discs look like? They are where one cardiac muscle cell ends and the other begins. I'll explain this more later in lecture, but it looks something like this, where one cardiac muscle cell ends. Notice how the nuclei are in the middle. These are unfused cells, but they act like they're fused because of these intercalated discs. And it's like a hash mark perpendicular to the long axis of these stacked tubes. And in this picture, you could see it here, and here, and here, even though this one's a little bit bent, here, here, down here. Those are intercalated discs. Now you can't see all of them, but you can see enough to know that that's cardiac muscle. Okay? And then we have nervous tissue. Again, there are no tissues in nervous tissue. If the question asks for cells, you're in tissue type number 4, nervous tissue. Because you either have a cell that conducts nerve impulses, and I will use what we call, there are different types of neurons, but I'm going to use a multipolar neuron, which looks like a large star. If you see a large star-shaped cell, that is a neuron. And the small cells that surround it, those are neuroglia cells. You can just call them neuroglia. Okay, we're getting down to the bottom of page 5. Now, if you look at this picture, this is again how you use critical thinking. You could say, well, look, it looks like the cells are separated by stuff, right? Except my question wasn't, what is the name of this tissue? My question was, what is the name of the cell? Boom, you go to tissue type number 4. Do you see how to use critical thinking? I'm teaching you how to use critical thinking so that someday you might be able to save my life with critical thinking. Okay? All right, it's the 2 cells, and the last 2 on page 5 are structures. The last 2, and I would write this on the bottom of page 5, the last 2 things is from a cross-section of a spinal cord. And when we take a spinal cord and cut it in cross section, we see in the middle what looks like a gray butterfly, like that. And I would call it a gray butterfly. I would always call it a gray butterfly because that would remind me that is gray matter. And the lighter stuff around it is white matter. Surprisingly, when we get into the brain, the orientation switches. The gray matter's on the outside and the white matter's on the inside. But I'm only for this lab quiz going to use the spinal cord in cross section, and the gray butterfly is in the middle, white matter is around it. Any questions on page 5? Okay, I have some websites. I have not checked it in a while to see if they're still good. If any of these websites go bad, let me know. The last one of these 3 is a dichotomous key, and a dichotomous key is a series of yes, no questions, and there's only 2 possible answers. That's why it's called dichotomous. You either go this direction or go that direction, yes or no. And you don't have to worry about that last link because I already ripped off the figure and it's right here. Where should we begin? I don't know. I think we should begin where it says start here. Okay, now look at that first question and look at how I told you how to start your 20 questions. A dichotomous key is a systematic way of doing your 20 questions. Look at this first question and tell me out of the 4 tissue types, which tissue type is this question designed to tease out and separate? Someone tell me.
The connective.
Connective tissue, because that's the only one where the cells aren't close together. And there it is. If the answer is no, you're in connective tissue. Second question in this one is separating out blood. Do you see a bunch of red circles without nuclei? Then the next one is separating out cartilage and bone. They're the only 2 with lacunae. Next question is separate. Do you see a cut tree trunk, concentric ring structure? If that's bone, yes. If it's yes, it's bone. No, that means it's cartilage. Hey, look, the last questions it's asking is about the fibers, just like how I did it. Okay. If the cells are close together, then it's the other 3 tissue types. This first question, that's separating out epithelial tissue. It's the only one with a free surface and basement membrane. Then it's going to ask about simple or stratified. Then it's going to ask about the shapes of the free surface. Everybody get how this works? You don't have to use the dichotomous key. You just have to mentally understand you've got to be systematic with your critical thinking. Then this thing becomes a real easy exercise. Okay? Now, what we're going to do now is usually this is where I break up the lab to actually look at glass slides on the microscope, and you're not here, so you can't do that. So what I would do if I was you is I would, for the next, let's say 20 minutes, hang out for 20 minutes, open a folder on your device, do a Google image search, and put pictures from the internet of the tissues that you're going to type in from page 5. Type in Google image. Type in simple squamous epithelium. Look for it and see if you could see a good picture of epithelial tissue with a free surface and basement membrane, 1 cell layer thick and flat. Then just go right down the line for the next 20 minutes. Just find as many pictures, maybe just 1 picture of each, but just find it, put it in the folder, okay? Then in about 20 minutes, let's say at 2:40, 2:45, I will go ahead and, because I did the same thing while students were doing microscopes a couple semesters ago. That's exactly what I did. And I just filled up this folder, and I will use those pictures I got that 1 semester in, again, about 20 minutes, and we'll play 20 questions. And I'll show you how to do this. Everybody got it? Okay, I'm going to go ahead and pause the recording and go ahead and do your own Google image search for about.
Your file of older pictures available so we can use them as reference as well as the pictures we have collected? Because if you say your pictures are perfect to practice with because they have everything that we're looking for, wouldn't that make it the perfect practice tool for us as well?
I don't have them labeled though, is the only thing.
More practice for us.
Yeah, I guess you can label it yourself. Yeah, I might be able to do it. I am going to have to quit the Zoom meeting at 3 because of, do you guys know that the technology department here, I am a legend of pity and sorrow for all the technology stuff that has gone wrong.
You're an IT nightmare.
Uh-huh. And so they're going to try to switch out my laptop so I can use my Bluetooth. Because, you know, if you notice in lecture, when I move away from the camera, my voice trails off. And then when I get close to the camera, it's really loud. Well, if I have Bluetooth, it'll be a constant volume. But this laptop that I'm using doesn't have Bluetooth, even though it looks like it should. That's my luck. So they're going to come here at 3 and take away this laptop. That's when I got to be done. And then while they're doing that, then I'll try to post the pictures on Canvas. How's that?
That would be perfect.
Okay, that'll be probably in a folder somewhere under files. Okay. All right, let's go ahead and look at some pictures. All right. Right about where that number 3 is, I want you to tell me what is the name of this tissue. Do you see a basement membrane and a free surface? And you're going to have to look carefully.
Columnar epithelial tissue.
Oh, you jumped the gun. This is the basement membrane here. This little slit right here in between what are called intestinal villi is the free surface. It is 1 cell layer thick, simple. All these cells are taller than they are wide, columnar. And here's the hint that I told you about. All the nuclei are in a row. This is simple columnar epithelial tissue. Okay. Let's see, hold on just a second here. How about okay. Right here. What is the name of the tissue? Do you see a basement membrane and a free surface? Whenever you see something circular, there's a good chance the hole in the middle is the free surface, like I showed you in the PowerPoint. But I'm talking about this one. Here's another one of the same tissue. So do you see a basement membrane and a free surface? You might not see the basement membrane, but this hole is definitely the free surface. This is lining the inside of a tube. How many cell layers thick is these?
Simple. It's a simple.
Simple.
Cuboidal.
There's one cell layer thick is simple. Are these as tall as they are wide, or are they flat? Look at that one.
Flat.
Flat. This is simple squamous. This is also showing, this is from a kidney, that kidney tubules that are simple squamous run right next to simple cuboidal epithelial tubules. So this is simple cuboidal epithelial tissue. This is simple squamous epithelial tissue.
So for example, for that picture, you would have circled which one you would want identified.
It'll actually have an arrow pointing straight at it.
Okay.
All righty. Do you see a basement membrane and a free surface? This one's easy. Right here's a basement membrane. You can see that this is a lining tissue. Basement membrane where these stained nuclei end. Up on top is a free surface. Tissue type number 1, epithelial tissue. Is this stratified? Sure looks stratified, don't it? How about at the free surface? What is this?
Columnar.
Columnar. Must be stratified columnar, huh? Look on the top. Look on the top of page 5.
Pseudo-stratified. Stratified.
Boom. This is pseudostratified ciliated columnar epithelium. You got to remember there is no stratified columnar on page 5. If you're tempted to write that, you got to know what's on page 5. This is your best friend. You got to use it. Also, the big hint is number 1 right here. Here's your cilia. Everybody got it? Okay. Let's do one. That might be even a little bit more fun. Do you see a basement membrane and a free surface? Yes, tissue type number 1. Basement membrane's down here where the stained nuclei come to an abrupt end. Free surface up on top. Is it stratified? Yes, it is stratified. That's the yellow word. What's the purple word?
Columnar.
Wait a minute now. We got squamous right here. We got cuboidal right here. We got columnar over here.
Is it transitional?
This is transitional epithelium. Transitional epithelium is stratified. So yeah, you might put the yellow word on there. When you go for the purple word, you'll realize there is no purple word. All right, do you see a basement membrane and a free surface?
No.
Are the cells separated by stuff?
Yes.
Tissue type number 2, connective tissue. Do you see lacunae? I don't see lacunae. Do you see a mishmash of cells?
Yes.
Splot. And a mishmash of pink and black? Splot.
Connective tissue.
Loose connective tissue. But you're not even thinking that until you realize you're in tissue type number 2, connective tissue. So you think these pictures will help you guys?
Yes.
All right, do you see a base membrane and a free surface? Well, you know, this is questionable up here, but I certainly don't see a basement membrane. These cells are touching, but they're not stacked tubes.
It's type 2.
It is connective tissue. This is the exception of connective tissue. The only connective tissue where the cells touch.
It's adipose.
This is adipose tissue, which are extremely thin-walled bubbles. Wouldn't you say these are extremely thin-walled bubbles? Kind of just like what I said, isn't it? All right, how about this one? Let's see. I don't like that one. Okay. Do you see a basement membrane and a free surface?
No.
No. Do you see a lot of parallel lines?
Yes.
Now, there's two types of parallel lines, one with very few nuclei, which would be because the cells are separated by stuff, or you'll see a lot of nuclei, stacked tubes. Do you see a lot of nuclei?
No.
No, the nuclei are separated by stuff. This is dense, regular connective tissue. All right, let's do this. Do you see a basement membrane and a free surface?
No.
No.
Are the cells separated by stuff?
Yes.
This is
That
connective
Highland
tissue. Connective tissue. Do you see halos around the cells?
Yes.
Those are lacunae. Are they in a cut tree trunk?
No.
No.
It's blood.
This is cartilage. Do you see black disorganized fibers, parallel pink fibers, or does it look like it's in purple jello?
Purple jello.
This is Highland
Highland
cartilage.
cartilage.
Oh, it's cartilage.
Yeah, see? This is doable, isn't it?
Yes.
All right, let's do another one here. Okay, this is going to be a little bit more challenging. Now notice how I'm asking the same questions in order. This is what it means to be systematic. Do you see basement membrane and a free surface?
No. Well,
Do you see cells separated by stuff? Yeah, I see cells separated by stuff. Do you see halos around those cells?
yes.
Is it
Yes.
in a cut tree trunk?
No.
No.
This is cartilage. Do you see black fibers?
No.
Do you see roughly horizontal parallel pink fibers?
Yes.
Yes.
This is fibrocartilage. All right, just a couple more here. This is one of the few ones you could probably jump a few steps. You see a basement membrane and a free surface.
Isn't that bone?
No.
The cells are in those little black splotches that are separated by stuff.
Bone, isn't it?
Do you see cut tree trunks?
Yes.
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 This is bone. What's this?
Blood.
Blood.
Blood. Dominated by pink circles without nuclei. You see one white blood cell here with a nucleus. You see these little fragments we call platelets. These cells are separated by liquid stuff. It is a connective tissue. Okay, I got a few more minutes here.
I have a question.
Uh-huh.
Should we write bone tissue or just
I went over
bone?
this. It's in the recording.
Okay.
4 letters, bone. That's all you
All
need.
right.
Do you see basement membrane and a free surface? You don't have enough nuclei to be stuck together to have an end that would look like a basement membrane.
I see tiger stripes.
Are these cells separated by stuff or are these stacked tubes?
Is that cardiac?
No, don't jump. You got to be systematic. Are these cells separated by stuff or are these stacked tubes?
Stacked tubes.
Stacked, they're in tissue type number 3. Do you see nuclei shoved off to the side, peripheral nuclei,
Yes.
with obvious striations?
Yes.
This is skeletal muscle. Everything that I told you was going to be in these views is in these views. This is kind of pixelated a little bit. Do you see a basement membrane and a free surface? No. Are these cells separated by stuff or are these stacked tubes?
Stacked tubes.
Stacked tubes. You see a few nuclei being in the middle. You know this is not skeletal muscle, even though you see subtle striations. So there's 3 muscle types with stacked tubes. The one that has these black lines are called what?
Cardiac.
Cardiac
is-
muscle.
Cardiac muscle. Those little black lines are intercalated discs. Almost done. This laptop doesn't like some of these pictures. Come on. There it is. Okay, this is a good example of like why you gotta be calm, critical thinking. What is the name of the cell?
Neuron.
Now you know you're in tissue type number 4 because of the question. This is a big star-shaped neuron. What is the name of the cell? Right over here.
Neuroglia.
Neuroglia. The small one's off to the side. Last one. What is the name of the structure?
Well, that's a spinal.
Structure is going to be the last 2.
White matter.
This is white matter. Does everybody see the gray butterfly?
Yes.
Yes.
That's gray matter. This is white matter. Everybody got it? There's certain little keys you've got to recognize. Then direct your thinking. Okay? Are there any questions for the first lab? Next week, Next week, we will go to page 6.
I have a question.
Yes.
What is the correct way to name the muscle and nervous tissue? Would
What's
you
that?
What would be the correct way to name the muscle and nervous tissues? Would you just
Oh, for tissue type number 3, it is 2 words: skeletal muscle, cardiac muscle, smooth muscle. And just what is written there for tissue type number 4: neuron, neuroglia, gray matter, white matter.
got it? Thank you.
Any questions?
Yes, one for me.
Okay.
You said that the test on Thursday will be going over both pages 5 and 6. Are we going over page 6 in the lab before the test?
We have our actual lab quiz in 2 weeks.
Okay.
Well, next week we cover page 6. The following week we have the lab quiz. It's all on
Okay.
the lab schedule.
Got it.
But
I
we'll
misunderstood.
actually have our, about this time, around 3 o'clock next week, will be our fake
Yeah, our fake
test
practice.
quiz to go through Proctorio.
Yeah.
Okay?
So
And-
our lab quizzes and exams are all going to be proctored through Proctorio.
Yes.
Okay.
Any other questions? All right, you guys, we got through week 1. If you have any questions, email me. I'm probably not, I usually answer emails on the weekend. I most likely won't this weekend because it's my birthday and I'm going out of town.
Happy birthday.
Happy birthday.
Happy birthday.
All right, you guys, have a good one.
Thank you.
Thank you. Happy
Bye.
birthday.
Thank you.